Mr. Azhar Tunio passed MSc Biotechnology in 2010. He enrolled in M.PHIL Biotechnology in 2010. Then he registered in M.PHIL in 2015. His research topic is antioxidant, antimicrobial, and biochemical investigation of some filamentous algal species from adjacent area of JAMSHORO. He is working under the supervision of Dr. Syed Habib Naqvi Saab and co-supervisor of Dr. Abdur Rasul Abbasi Saab and also co-supervisor of mine. Now I invite to Mr. Azhar Ali Tunio Saab is come and deliver first M.PHIL seminar.
Start with the name of Almighty Allah, who is most merciful and creator of entire universe. Prestigious Professor Dr. Abdur Rasul Abbasi Sahab, Dean of Natural Science and also my co-supervisor. And respected Professor Dr. Syed Habib Naqvi, my supervisor, and co-supervisor Dr. Khalid Hussain Lashari Sahab, and teachers and all of you, Assalamu alaikum. As you all know that today I have invited here to present my first M.PHIL seminar. The topic antioxidant, antimicrobial and biochemical investigation of some filamentous algal species from adjacent areas of JAMSHORO. So as far as my little knowledge concerned. Content. Introduction. Algae. An algae is mainly aquatic, eukaryotic and photosynthetic lower plant organism. They can grow rapidly and live in harsh conditions due to their unicellular or multicellular structure. Algae are used throughout the world and a very large diverse group of simple, typically autotrophic organisms. Algae unicellular to multicellular forms such as giant kelps, diatoms that grow 65 meters in length. Algae have a great potential for developing novel biotechnological applications to underpin building a bio-based economy. It is estimated that 50,000 species exist, but 30,000 have been studied and analyzed. Algae consists the chlorophylls bearing plants, thalloids, and autotrophic and aquatic. Habitats of the algae: aquatic, freshwater and marine, example moist stone, soil and wood. Association with fungi, example lichens, and association with animals, example sloth bears. Marine forms are seaweeds. Freshwater algae microscopic unicellular, example chlamydomonas. Colonial forms, example volvox. And filaments forms, example Spirogyra and Eulothrix. Applications. The application of the use of microalgae for human consumption as a source of high value health food. Macroalgae and microalgae have a broad range of biological activities such as antiviral, antimicrobial, anti-inflammatory, anti-carcinogenic activities, etc. Antioxidant compounds used to protect from the free radical during oxidative stress in the cell. Compounds can be derived from natural source. Natural sources are safer to use due to less toxicity and side effect. Algae as a nutritional and energy source, and algae is used as food, agar, manure, and also used in nitrogen fixation. As well as in pharmaceutical industries, because algae also contains some medicinal elements. Algae used in methanol production, ethanol production, biodiesel, power plants, and cement plants. Microalgae are prokaryotic or eukaryotic photosynthetic microorganisms that produce carbohydrates, proteins, lipids, and as a result of photosynthesis. They provide many vitamins, includes vitamin A, vitamin B, B2, B6, and vitamin C, and rich in iodine, potassium, iron, magnesium, and calcium. Selected algal species: Oscillatoria limosa, Microspora stagnora, Spirogara equinoctus. These three species collected from the different three stations adjacent areas of the JAMSHOROs. Here is the classification of these species. Oscillatoria limosa, filamentous in the colonies, cancelled is a light source, commonly found in watering troughs and water that is mainly blue green or brown green. Its genus Oscillatoria and order Oscillatorius are filamentous cyanobacteria with which is named for oscillation in its movement that produced by fragmentation form long filaments of cells which can break into fragments called harmogonia. The aims of objects of three different species. The aim of present study is to evaluate the potential of antioxidant activity in 20% extracts such as acetone, methanol, ethanol and aqueous of selected algal species. Some biomolecules metabolize total phenolic and phenolic content. Will also analyze from acetone, methanol, ethanol and aqueous extract of under study collected algal species. Correlated the biomolecule with antioxidant, antimicrobial activities as well as a polyphenolic compounds. Material method. Algal species. These are the three different stations from where I have collected the Oscillatoria, Limosa, Microsphaera, Stagnorum and Spirogyra equinoctialis. The three different stations of agent areas of JAMSHORO, I have collected these species 5 to 7 times on the monthly basis during the time AM to 12 PM. pH, water and environment temperature were noted at the spot during the collection of the samples. Preserved in 4% commercial format for taxonomical studies as the cells can survive their lives. Algae were identified with the help of microscope on the basis of their morphological characters. Selected areas of study. Station 1, species collected from pond near Wabda office, JAMSHORO. Station 2, species collected from JAMSHORO to Koti Road near Court. Station 3, species collected from pond near Honda Palace near Bypass Bridge, Hyderabad. Here is the preparation of the extracts and 4 solvents. Preparation of 20% extract means 20 gram of dried algal sample cursed with the little amount of different solvents, ethanol, methanol and water. Centrifugation. Here is the 2 times centrifugate at the 15 minutes at the 16,000 RPMs. Filtration. After that I filter these three species with the filter paper's help. Finally take up the volume 100 ml. And after that I have measured the pH of different samples and solvents. Furthermore will be biochemical analysis. Here is the different species from I have collected the different and also with the help of co-supervisors. I have collected these series species and maintained the water temperature, environmental temperature, and at present time I have measured the pH. Results and discussions. pH of water stations during the sample collection. But the highest pH noted in the station 3, Spirogyra equinoctialis species, and the lowest pH found in station 2, Microspora stagnorum. Water temperature of different stations on spot. The highest water temperature around 32 temperature was measured and lowest water temperature measured 29 at on spot. Environmental temperature of different stations. Environmental temperature around 40 measured in Spirogyra station 3 and lowest station 2 Microspora stagnorums. pH of different algal extract in station 1. Here is the only one species, the species that is that is prepared agent in 4 solvents like as water, ethanol, acetone, methanol. But the highest pH found in acetone and the lowest pH found in water samples. Here is the station 2. Here is the station 2 and made test in the 4 different solvents: water, methanol, ethanol and acetone. But the highest pH found in Here is the only one species of the station 3. They made test in the 4 different solvents: water, ethanol, acetone and methanol. But the highest pH found in water sample and lowest found in methanol of Spirogyra equinoctialis species. Here is the final calculation of these three different stations and these 3 species. Oscillatoria station 1 made test in 4 extracts: water, ethanol, acetone and methanol. But the highest pH found in acetone of Oscillatoria species. And the second green color shows that the highest pH found in methanol found in the station 2, the Microspora stagnorum species, and the lowest found in water, Microspora stagnorum of species. The third one, the Microspora species, highest found in the water and the lowest pH found in methanol extract. The final calculation tells that the highest pH found in acetone of Oscillatoria stagnorum species and the lowest pH found in the ethanol samples of Spirogyra equinoctialis species. Conclusion: it has been analyzed that 3 stations of algal species shows different temperature of water, environmental and different pH noted of each species. Highest water and environment temperature and acidity examined in station 3. During the sample and low water and environment temperature recorded in station 2. 4 extracts were acetone, water, methanol and ethanol of algal species shows different pH, but the highest acidity found in acetone extract and low pH and acidic condition station 3 in methanol extracts. Future plan: different biochemical metabolites like total sugar, reducing sugar and total protein. Antioxidant, phenolic, flavonoid compounds will be analyzed and quantified from different identical algal species by reported methods. 3 amino acids and sugar will be detected by thin layer chromatography. Acknowledgement: I am humbly thankful to Almighty Allah who gave me the potential to write this manuscript. I am thankful to my supervisor Dr. Habib Aman Nakvi, Institute of Biotechnology and Genetic Engineering, and co-supervisor especially thanks Professor Dr. Abdul Rasul Abbasi and my co-supervisor Professor Dr. Khalid Hussain Lashari, in the shore of Allah's blessing with the moral support of teachers Dr. Muhammad Umar Dahood, Dr. Muhammad Rafiq and Madam Komal Siddiqui, and teaching and non-teaching staff FG at University of Sindh. My parents, classmates, friends, I become able to contribute this little knowledge contribution in the vast field of knowledge. Here's the references. Thank you for the concentration, and further the future plan will be analyzed in these solvents.
Or maybe some diseases which convert into the thalassemia. But I have also seen a lot of children are suffering, and really very upsetting situation, not only the Pakistan but all over the world. So very interesting, and inshallah you will do most work on this project. And at first this stage I will advise you to collect the literature information about these hemoglobinopathy diseases, what is actually the reason of this shortage of the level of the hemoglobin, and why this hemoglobin level is changing into thalassemia. So anyhow, other projects which were presented, they are also their own importance, and the audience was very silent because they did not take much interest in asking the questions or other things. Anyway, the seminars, they were the first seminars, all of them, and they were very good in first seminar presenting here. Therefore, I approve all these 3 seminars. And inshallah, in next seminar, they will be with very fruitful and with nice results, inshallah. And I also appreciate the efforts of the supervisors, and I congratulate them and I feel they helping me, because I was very interested to encourage the researcher. And if you see, maximum number of the scholars are admitted or enrolled during my this tenure, and I hoping some more as well. But sometimes the higher authorities are not with me, but anyhow I will try my best. And the university really is for the research. These BAs are, this is the function of the campuses or the colleges, and the universities for research. So thank you very much, all of you. You have been here with me. Thank you all.